we are able to go equivalent of that house level within the brain, which is at cell level, but across the whole brain.
It sounds simple to say imaging of a human brain.
Yep.
But it's a huge engineering challenge.
Yes. Currently, the largest, most detailed brain map of the developing human brain in second trimester.
So the Dharani dataset for each brain will be able to track these 80 billion odd neurons.
It's in the largest collection in the world.
Hi, this is Amrit. We are at IIT Madras, my alma mater, and India's top university for people who like to build. We are here to meet some builders, ask them, what are you building? What does it take to build? And what makes IIT Madras the best place to build?
Hello and welcome to the Best Place to Build podcast. Today we have with us Dr. Richa Verma, Chief Scientific Officer of the Sudha Gopalakrishnan Brain Centre at IIT Madras, which does brain imaging. We'll learn about it. It does brain imaging at 0.5 micron level or cellular level. The research group has a wet lab, an engineering lab, an optics lab, an imaging lab, a software team and an analytics team. So it's a large team, multidisciplinary. Dr. Richa, welcome to the podcast.
Thank you, Amrit. Thanks for having me here.
Doctor, I have to ask you, what is brain mapping? What is brain imaging and why do we need it?
Okay, that's good and something good to start this conversation with. When we talk about human brain or any brain for that matter, it's an organ which is very heterogeneous, which means it has different cell types, there are different regions. So to understand the brain, you want to know how these cells are organized and how each of these organizations makes different brain regions. So just like world map, you identify different countries, states, towns and cities, and you then create maps, right? And then you put different countries mapped together to create the big atlas. If you remember in your school, you would have in geography the atlas, which would comprise maps of different countries. So same way, we are creating human brain maps where we have detailed information about different brain regions. And when we put all of this together, what we are doing is generating brain atlases.
Nice. You gave me the analogy earlier of moving from an atlas to something like Google Maps.
Yes, or the other way around.
Right. So,
okay.
So in the sense, what I want to understand is, I mean, in the introduction, I read that it's at a 0.5 micron level. How much does the impact of the resolution matter?
Okay. So the closest analogy, which works with a lot of people, we have had world maps for centuries, right? People used to navigate from one country to the other. And those maps have also evolved over a period of time. Now, if you compare the world map 200 years ago versus what you see in Google Map, Google Map, you're able to go to the street level, to individual house level, right? So what we are generating in terms of human brain maps, we are able to go equivalent of that house level within the brain, which is at cell level, but across the whole brain. And that is super, super critical. The reason being, you want to understand several brain diseases, different developmental disorders. And if we don't understand the organization of each of these brain regions in normal brain, it's very hard to crack the problem when it comes to diseases.
Very interesting. I've read somewhere, I mean, I don't know where, but you get all this information from different places.
Yeah.
That a certain kind of activity lights up certain kind of the brain, or certain kind of diseases are caused when certain parts of the brain are not functioning properly. So are you saying that a detailed brain map will go much more detailed in saying that this part of that part and at this address in the brain there's some malfunction?
Yeah, so as of now what we are doing is, when you create these normal brains map at different age groups, and we are doing it from prenatal stage, that becomes a reference maps. So if you are looking a brain at, say, in third trimester or second trimester, you have created a very detailed map of a normal brain. Now that can be used as a reference to look at different disease conditions, or in this particular case, developmental disorders, and see which region does not match with this particular normal brain. What are the cell types which has been affected? Are there numbers which are less, or the overall brain thickness which has been, or what we call as the gray matter thickness that's been compromised, etc., etc. So hence this is super critical when I said that, you need these reference map across the entire age group within the developing human brain, but also as we are born all the way up to when we age. Okay, and this is what we are doing.
Okay, so at the very simplistic level, the first level impact of your work is that we will be able to compare two different brains, find out what's different, and associate the manifestations of that difference onto some causative or some correlation in the
cause, and then look at what has really got changed in this particular disease condition, in which region, and then you investigate that further.
Nice.
Yeah.
Okay, so I get that. And we'll go into detail in a bit. But before that, with this background, can you tell me what the Brain Centre does?
Okay, excellent. So the overarching goal of the Brain Centre is to map the whole human brain all the way from prenatal stage to 100 years. And very happy to share that this work started 5 years ago, but the physical center, the Brain Centre, is now 3 years old. And we have been successfully, mapping different types of brains. And one of the data which we released early this year is from the second trimester developing human brain. And that's called as Dharani. It's an open source
Dharani.
Dharani, Dharani. And that's currently the largest, most detailed brain map of the developing human brain in second trimester. More importantly, the data has been made freely open access data sets. So if you go and browse Dharani Atlas, you can access the entire data set. So so when we started this work of brain mapping, we did not limit ourselves to only look at the normal brains. In parallel, we have been studying the developmental disorders. We have been looking at brains affected by stroke or ischemia in specific, what we are currently looking at, neurodegeneration, but also mapping the aging brain all the way from 50 to 100 years old.
Interesting. So many questions come up. In my mind, I'm wondering, I met Professor Mohan on the podcast. He had come last year sometime. And he said that it sounds simple to say imaging of a human brain.
Yep.
But it's a huge engineering challenge.
Yes.
Can you explain to us why it's an engineering challenge?
Okay. So if we look at what is currently, how do we image human brain? Especially I'm talking here in living human brain. Most of you would be familiar with MRI or a CT scan, which is often used to look at different diseases, different conditions in living human brain.
Yeah.
And, in the last 20, 30 years, that has been, the impact of doing MRI has been quite profound. You can look at human brain and study where the problem is. The problem with this sort of imaging is the resolution at which you can see the entire brain is at the best at 1 millimeter. Now, 1 millimeter, you will not be visualize the different cell types within the brain. If you look at the basic science research, specifically in animal work, and I used to work on monkey brains, there you can, you can do many things, many type of intervention to study the whole the whole brain. But the big gap in terms of when you look at postmortem brain, people often study small regions or small areas within the brain at cell level. So what we have developed here is closing the gap between the volume and the cellular resolution. And what that means is, we are working on postmortem brains. We get these postmortem brains in a very good condition from our clinical partners, of course, after the written consent. Once we acquire these brains, we also do a postmortem structural MRI, where you get a reference of the brain region inside the skull.
Hold on.
Okay.
You're going too fast for me. So
All right.
I need to dial back. So I did some studying before coming.
Yeah.
And I understand that I have not had a brain MRI.
Yes, me neither.
But I have seen it on TV.
Yes.
In these medical shows.
Yes.
Where they show these black and white sort of images which you put on against a light.
Correct.
You read that. So so the MRI is at 1 mm.
Best. You can get a different resolution.
And in that you can see what?
You can see, I would say, different layers, the different brain regions. You can make interpretation about the vasculature, etc. There are different protocols.
Okay.
But you cannot look at the cell level or what is the neuron level.
Okay.
That's not the resolution of the clinical MRI, what we currently have.
You're also using a word volume. What does that mean?
Okay. So when you do what we call as histology, you take small portions of any tissue and you slice it, and then you use different chemicals to stain depending on what you want to investigate in that tissue. And then you can put it under the microscope and look at the detailed cell level. Now, when normally we do this, we are limited by small regions. Ideally, one would like to see the entire brain. An adult human brain is close to 1200 cc to 1500 cc. You want to see the entire whole human brain at cellular level. So your MRI imaging does not give you that.
You're saying it very casually. How many cells are there in this 1200 cc?
So as of now, the best known information is 86 billion neurons.
86 billion
neurons are there in the adult human.
So the Dharani dataset for each brain, I will be able to track these 80 billion odd neurons.
Yeah, so first thing, that Dharani dataset is the developing human brain in the second trimester. But yes, we are working on really estimating the total number of cells at different ages across the entire lifespan.
Okay,
yes.
That's fantastic. Yes. Thank you so much. I got a sense of that. So you were talking to us about the engineering process of doing this imaging.
Okay, yeah. So
you have with consent, you have
we got the brain.
Postmortem brain.
We did the structural postmortem MRI.
Okay.
Then the brain has to be, and this is a biological tissue, which is soft, but also has a very high water content. And the goal here is to slice this 1200 cc or 1500 cc biological tissue in very thin sections, as thin as 10 micron or 20 micron. To do so, there are different steps which, is normally used. And what we do with these tissue is we put it.
What do you mean by normally used? Is this a?
So, this type of work has happened before.
Okay.
It's just that it has not happened at this scale.
Okay.
And it has not happened at this speed and this resolution for the whole brain.
Okay.
Yeah. And what we use here is we actually first take the water out of the brain, and we call that, cryoprotection.
Okay.
Because we are going to freeze this biological tissue. And once the water is, replaced by sucrose solution, we then freeze the entire brain at minus 80 degrees centigrade. Okay,
so first step is postmortem MRI. Second step is cryoprotection. Third step is freezing at minus 80. Okay.
You got it right. And now once you have, got this frozen brain, it's hard enough for us to really slice it at 10 or 20 micron thickness or even 50 micron thickness, depending on what you want to achieve.
So the fourth step is slicing at 10 micron.
Yep.
And how is this done?
So in a traditional method, people would you will use a machine called cryostat. You
Okay.
slice it and you lift a tissue section using a brush and then float it on a particular solution. What we have developed here is a tape transfer technique, which comes with a specific glue that allows you to lift, because here, we are talking very large section, close to 6 inch by 8 inch. And that particular section is 20 micron thin. So imagine lifting such a large section at such thin slices. We use this specific tape, which comes with the glue.
Okay.
So the tissue sticks on that glue. You lift that tape and the tissue together. You then use a UV light to uncure it and transfer this thin slice onto a glass slide.
Okay. So, and after the slicing, there's a transfer.
Yep.
And can I ask you, here there's a clear innovation that the method that you've developed is different from everything else. Is there, like, at every step, have you had to innovate?
Many things. So even when I spoke about freezing such large tissue, that has not been done. The reason is people have often worked on animal tissue or human tissue. They slice it much thinner so that you don't have to worry about a very large volume of a biological tissue to be frozen at minus 80 degrees centigrade. And if you think about it, that's a very specific process, because tissue is, still has a lot of water in it, even
So it
though
can expand.
tissue can expand, and depending on if you go too fast, it can just crack.
Yeah.
If you go too slow, you will have these freezing artifacts. So we had to optimize that, and that took a while to be really able to freeze such large organ. And
I can imagine that, like, if it's about this big, right?
Yes.
So if it starts freezing from the middle or starts
yes,
freezing from
exactly, you will end up seeing the artifacts, and that's of no use to us because we want to image the whole human brain at cell level. So those cells just start bursting, and that's, that's pointless for the effort what we are trying to achieve here.
And I guess if that does happen, you will see it at the end. You
No,
may
when
not
you start
realize-
slicing, you will see it. But
Okay.
now the way we have optimized, doing all the experiments, the rate and the time it took, to freeze, we have a very good idea that if the freezing has gone well or not. And to be honest, we have done this now across 100 plus brains.
Okay.
And this is quite a standardized process in our lab.
I can understand now why you have such a multidisciplinary team.
Yeah.
So, postmortem, cryoprotection, freezing, slicing, transfer, and then?
Transfer. So now, if you come to a lab, or rather if you look at these brain tissue, as soon as they are sliced, they are very transparent, or if I can say translucent, whitish color. You can't really see the different cell types what you want to study. So the next step is to do chemical staining, to study different cell types, different protein markers, etc. Now, based on the type of brain we are studying, what we want to investigate, we have 2 or 3 standard staining, but then we add different protein markers to ask specific questions.
Okay.
So then each of these brain slices, which have been placed on glass slide, goes through the staining process. That has, each one of it has different protocols, different timing, it's stored at different temperature, then it's allowed to dry. Now, once it's dried up, the next step is now to digitize these data sets. So if you think about, or if you remember, I don't know if you did biology in your school, you would have to go.
I will keep quiet on that question.
All right. You would have to go and look at tissue or anything in detail under the microscope.
Yeah.
So when I say we have to digitize this data, the scanners which we use, these are scanners which can scan large tissue section. Those are light microscopy technique, where now when you said, at the start that you're imaging at 0.5 micron. So those scanners allows us to image these tissue slices at 0.5 micron resolution. And we, have this entire whole brain data set which gets digitized. And the beauty of it is you don't have to go under the microscope to look at it. These you can access. We have built our own, inventory, and we have our own learning management.
But I want to ask you a question here. You have 10,000 odd slices.
Yeah.
Fewer for a fetal brain. I understand that.
10,000 odd slices for every adult brain, roughly.
Yeah.
Yeah.
So that is, chemically stained and then imaged and digitized.
Yeah.
But for me as a researcher to access your data set, these are 10,000 different images I'm looking at.
Yeah.
Isn't it like how do I read them?
So it's all very well. So if you want to, as an individual researcher, you might not need all 10,000 slices because you are interested, and this is how most of the neuroscience people work. People are working on specific brain region or specific cell types, etc. That's what they are interested in. But they have access to all of our data sets. We give them, depending on how big the data is and what they are looking for, we give them the access. They can then navigate through different modalities which we have used, different staining types. They have access to the postmortem MRI data sets, etc. And so if there was an option and I could show you how our viewer looks like, you will be able to understand this better.
That's fantastic. So maybe what we can do is that there's a tool that you've developed.
Yes.
We could, when we are editing the podcast, the editor could just add it as a
Yes. And I could show you. So specifically, the Dharani Atlas, which we made it as an open source, I can navigate through all the different modalities. So you as a researcher sitting any part of the world, how can you access and what type of data sets you have access to? Yes.
So if I'm a researcher in any part of the world, and say I'm looking at Down syndrome in third trimester kids, third trimester fetal brain, I could go to the Dharani Atlas website.
Currently, yes, it is second trimester, but if you want to see how a particular brain region is, in a normal brain, yes, they have access to all of that.
And I could just click saying open this.
Yeah, you can just click, you can zoom in, you can look at cell level, and you can then go through, you will be looking at the 2D image at one point, but you can go navigate through the entire series, and you can also look at the 3D data sets, how it looks when it's all put back together.
And I can compare this to a healthy reference.
Yes. And more importantly, we have also made it accessible in a way that if you need further information on any particular data sets, please contact us.
Okay.
Somebody would have much deeper questions, and we are quite open to it.
So the first step is post-mortem MRI. Second step is cryoprotection. Third step is freezing at -80. Fourth step is slicing at 10 micron. Fifth step is transferring it to slides, which you said that itself required innovation.
Yep.
Then there's chemical staining with different markers for different kinds of questions that you want to ask the image.
Yep.
Seventh is imaging it, digitizing it. The eighth step is that you upload it onto your software tool, which you have developed.
Yep.
So that researchers can access it.
Yes.
And the ninth step would be that if researchers are really keen to ask further questions, they could contact you.
Absolutely.
That is very intense. I want to ask you one question. Who are the kind of people who join your lab? What kind of backgrounds are they from? What kind of disciplines is required to do all of this work?
Okay. I am so glad, Amrit, that you are asking me this question. We often hear about interdisciplinary teams, and we need to, work across different disciplines. If you come to our center today, we have close to 120 plus people at the center. And this ranges from engineers, could be mechanical, electrical, computational team, a very big team. We have research associates from life sciences background, few with masters in neuroscience specific, but in general we have from different streams within the life sciences. We also have people with radiology background, people with image processing. We have several technicians or histotech. We also have people specifically working with scanning tools. And as I mentioned, this entire thing, what I just spoke about, are the people who are physically sitting here. But most importantly are also our medical partners, doctors who have been working with us now for 5 years to really acquire a very high quality post-mortem brain tissue. Without that, this entire effort would not have been possible. So today
they're giving you like contextual information and
no, so we do get the clinical or the preclinical data, etc., and that's from the hospital database. But also, doing and extracting this brain in timely manner is super important.
Right.
Because, the postmortem interval is very critical. If that gets delayed, the cells, even though those cells have been dead, but you want to sort of trap those cells the way it was when the person died, and keep it in a particular solution within certain time, so that you don't see much of deterioration. So they are the ones who don't sit with us, but we work constantly with them. Sorry. And then we have a whole bunch of our collaborators, both in India as well as overseas. And that is expanding quite a lot.
I want to just say, this is the first time you're using the word India, because earlier you had mentioned that this is the largest collection of fetal brains
yes.
And I was waiting for you to say largest in India.
No, it's in the largest collection in the world.
Okay.
The data set which we released, Dharani.
Nice. So medical partners, and you were talking about the partners who use
these data.
Yeah.
So as I said, this atlas got released early this year. We now have collaborators in India and overseas who have been looking at the developing brain in humans as well as in animal model, who are using these different data sets to query their questions, what they have been asking. And that's the first step. And as I said, because this is just 6 months old atlas, we have already got a lot of attention because, seeing a developing human brain in such detail has not been done before. And that's what is attracting a lot of collaborators who really want to use these reference data sets.
Can you give us a sense of who these collaborators are? Where do they sit? Are they labs? Are they
mostly
companies?
Are these labs, neuroscience people. So company is a different, that data set would be used in different context. But in terms of neuroscience research, we have people in INSTEM, Dr. Bhavna Muralidharan. She is working closely with us. We have collaborator in UCSF. We are now collaborating with people in Canada. So different groups across the globe. And I'm happy to share, or if you go on our website, the entire list of collaborators, which is close to 20 plus.
This is a post-mortem intervention.
Yeah.
Is it possible that MRI kind of techniques will develop to a level that this can be done while the person is alive? The head MRI machine is like one science fiction machine which sits like this, right? And you're saying that is a 1 mm resolution.
The clinical one. So people use higher magnetic strength for animal study. I don't know if you have heard. So in clinical setup, specifically in India, the highest magnetic strength is 3 Tesla.
Okay.
In animal studies, you can go to 11 Tesla, etc., which can give you at a very high cellular resolution. But in living human, putting a human through such a high magnetic strength, I don't think so that's going to happen.
Okay.
However, what we are hoping as we generate these atlases, you can overlay the cellular information on the MRI and then create different models to predict what you're seeing in MRI, what would happen at cell level. That's the big goal.
I want to just note here in my notes, it says that the inventor of the CT scan and the MRI both got the Nobel Prize. They were teams.
And the CT scan guy got Nobel Prize for something different than the CT.
Oh, is it? Okay, okay.
But don't worry.
Fine. Should have noted this down here.
Yes.
I have a question here, but before this, I want to ask you a question. All of this sounds really expensive. Like, does it how much, if you can share, I don't know if it's something you can share, but how much does it cost to take one brain through all of this process?
It is a very expensive affair in terms of all the process, the consumable, the chemicals, etc. I'm not talking about the equipments here. And we are looking at, in Indian rupees, each brain would cost you roughly, again, it also depends on the different protein markers which we are using, but if I can share, it's close to 2 crores per brain.
And we are not even talking of the center cost and the equipment
No,
cost
we are
and the
not.
people
We are
cost.
just talking the
120 people.
consumable.
This is a hugely expensive affair then.
Yes, it is. It is.
And you're saying that the entire data set is released publicly.
Yep.
So then my question is, who funds it?
Oh, we have a long list of funders, and I think in Mohan's podcast, you have got more of those detailed information.
Yeah, he
But-
talks about, of course, Chris Gopalakrishnan.
Yeah, so this work started with the support from Principal Scientific Advisor's Office, which funded the project in 2020 Feb, and the work started in March 2020. And of course, the Pratiksha Trust by Chris Gopalakrishnan. We have specific ischemia work funded by Prem Vatsa, who is based in Canada.
Who's also an alumnus.
Yes, yes, exactly. And since then, we have got a whole list of different fundings which has come through, and a lot of philanthropists who are really funding this work because they can really see the potential in what the center can do, and we are very grateful for that.
I think there are a lot of funds across the world which also look at furthering the understanding of diseases like Alzheimer's and
Yes.
dementia.
Yes,
I'm guessing that since the life expectancy of human beings has grown from like 40, 50, 100, 200 years back to like 80, 90, we are
yes.
seeing more and more brain conditions come up.
Oh, yes.
There's a lot of data that autism itself is probably something we'll learn more about
Yep.
if we know more about the brain. So I guess this is an area of research that is booming, right?
It is booming, and it is also booming for several reasons. So there are few organs in our body where we have understood it much better, like the heart. But when it comes to brain, human brain, we understand very, very less. Primary reason, the most of the neuroscience research has been on animal model. And there are limited things what you can do on living human brain. And the nature of the human brain, the way it is organized, as I said earlier, it is very heterogeneous. If you take the brain portion from the front of the brain versus the back of the brain, it's very different. So due to several of these factors, we have understood the brain quite well, but we are still far, far away when you really want to understand the organization, the functioning of the human brain. And hence, this particular field of research is what you called it as booming. But it's going to continue, and especially when we add the modern day computational tools, we are all hoping to address a lot of unresolved questions for decades. Because when you have these large data sets, you really want good AI models to be looking at these patterns, which, human eye just can't pick it up. So yes, this particular field is expanding at different aspects. And that is something which I can say for youngsters is a good place to be if you want to pursue a career in neuroscience or computational research within neuroscience group.
Yeah, I can imagine that. I want to ask you a little bit of detail here. This is very foundational research in the sense that
Yes, basic
we
science.
talk of foundation and translation research,
Yes,
and
yeah.
usually the criticism of research from Indian labs or IITs is that it's more translational. It's application.
Yep.
But this is not application research. This is
Not yet.
very foundational. This
Very
is at the opposite end of.
Opposite end, and we really want to go deep in that so that then you could have a very good translational effect. And that will happen as we generate more of these data sets, ask specific questions, and really go, one of the big thing in research is every step in the process, how you achieved it, rather than just focusing on what you want to achieve. And I think that's what we are aiming at.
So you are saying that if the foundational research was done at a very
Basic.
good understanding and detailed
Yes.
level, then
Yes.
all the translation research that's built on top of it will be
More robust, and you would have more meaningful results out of it.
I was thinking that if this is being done from India, then that also encourages other researchers who are maybe not as patient as you are
Yeah.
and who want to be more on the translation side.
Yeah.
They can build on the work.
Oh,
that you've
yes,
done.
we are. So, it's not like we are not looking at translational effect at this point. We are not saying we are going to wait for something to come out. We are engaging with a lot of neuropathologists, different doctors, and people who work in different disease conditions to come and look at this data, because even though you might be working in different animal models, but you have been asking a question, for example, on Alzheimer's for the last 30, 40 years, right? Now you have access to this sort of detailed human large brain data sets. We strongly work with those people so that, the translation effect happens much sooner than later. And that's the way to go. One of the things I do want to highlight, because when you said this is a booming field, we are working on human brain data sets. What we are doing is truly state of the art, not been done at this scale and this resolution before. At the same time, we are integrating data sets from other modalities and working with different collaborators. So when you want to understand brain behavior, you want to understand from genes to molecules to cell, all the way up to brain behavior. And everything one lab cannot do it, or one center cannot do it. This is common across the globe.
Yeah.
But when you integrate and work with different people, either they have expertise in a particular disease or a particular brain region or a particular age group or a particular model. They might be working on a monkey model or a mouse model, or they are working on, suppose, MRI imaging on human brain. When you put all of this together, that's where you're looking to have a really good translational effect. And that's the goal, the way we are collaborating with different people across the globe.
Understood.
Yeah.
In my mind, I'm thinking of when the human genome was sequenced like 30 years back.
Yep.
And that was a very concerted effort.
Yes.
And it created a bunch of, I don't want to use the word atlas, but
yeah,
a library of different
exactly
human beings' genes sequences. And that sort of catalyzed a lot of work.
A lot of work in last 30 years. Yeah.
Yeah. And when COVID happened, we had a vaccine that was an mRNA vaccine, which was clearly built from that step to that step. You could sort of draw a line. It's amazing.
Yeah, no, that's exactly why so many people need to come together to solve such a big problem of understanding the human brain.
A lot of teams will sort of have to come together across the world.
There is no other way out. Otherwise, you can keep, doing this sitting in your own individual lab for decades before you could really figure out any disease or any brain region.
Damn fascinating. Dr. Richa, this is, I want to ask you, what is your background? How did you land up here? And what is this journey that you have taken? And how can people take the same journey as you've taken?
Nice. So, going back, I don't know how far do I have to go back.
Why did you start as an undergrad student?
Yeah,
or a high school student?
No, high school, we'll skip it. But undergrad, I actually did it in Chennai, Shankar Nethralaya.
Okay.
I did my undergrad in optometry. And then I went to Australia to do my PhD at University of Melbourne in retinal electrophysiology. Retina, which is part of the eye. And that's sort of, motivated me to go deeper at cell level. And sort of the retina is the neural tissue of the eye, and that's connected to the brain. And that's where I started getting interested. And I did my postdoc at Monash University in Melbourne, working on monkey brain.
You mentioned monkey brains earlier. I wanted to ask you about
So
that.
I used to work on doing the electrophysiology, where we would do recordings from single neuron, but also then do the similar work, slicing the brain and looking at different brain regions within the monkey brain. And I was also looking at the retina. Then I joined as a faculty at School of Medicine, School of Optometry at Deakin University, where I was doing a lot of higher education, teaching, learning, etc. And that was a very good experience because, only when you teach, you learn more. And that was the time when we sort of heard about Computational Brain Research Centre coming up here. And I was in Australia for 14, 15 years. So it was a big step, taking this decision. So I did move here in 2017, mid of 2017. I was working with Teaching Learning Centre, IIT Madras, L.V. Prasad Eye Institute in Hyderabad. And happy that the funding came through and the Brain Centre work started. So when we started the work at Brain Centre in 2020, it was just 5 of us, including Professor Mohan.
Yeah.
Yeah, and today, as I said, we are looking at 120 plus people. So it has really been an exponential rise.
Yeah, you sort of gave us a little bit of breakup here, but Professor Mohan is the principal investigator.
Yes.
And Professor Jairaj is the co-principal investigator. They are both electrical engineering professors.
Yes, yes.
I'm always fascinated by the fact that Professor Mohan is an electrical prof who has such a high impact on the biosciences.
Yes, yes. So I think you would have, this, Mohan has he heads the another center, Health Technology Innovation Center, which is located at the Research Park. And that work, I think, if I'm not wrong, started around 2011 and 12. So he's very well connected to the medical fraternity field in India, developing technologies, etc., for a long time. Yeah. So I'm glad he got interested in the brain. And to process this whole human brain, we had to build a lot of equipments. So if you look in the market, most of these steps which I spoke about, staining, sectioning, etc., most of it is designed for small brains or small portions of human brain. So we had to develop a lot of technology and equipments. And if you have visited our center, you will be able to spot most of those equipments were all built here, R&D in progress, but we have been using for the last 3 years. So that's where the entire engineering experience comes in picture. And then once the data starts coming out, the whole computational team is now expanding because we are building very strong analytics. A lot of youngsters are joining our team, trying to build different AI models to understand the brain. And as I said, this is a tougher problem because the brain is very heterogeneous. If your model works very well in this part of the brain and a small
it may not work
portion of the tissue, it will not work there, and it will not work so easily from one brain to the other. Yeah, so
thank you for sharing so much. I just want to note that looking at your career and Professor Mohan's career, and I'm sure the other people,
many more people,
maybe you can tell us a little bit more. I think you have a collaborator called Dr. Jay.
Yes.
I want to hear about his career also.
Yes.
But I want to make this comment. At a UG level, maybe less at a PG level, but at a UG level, everybody is too stuck about the degree that they get. and I can see that you started with the I.
Yes.
And there's so much movement, right?
Yep.
And for me, that's so obvious. You cannot stick to the first year course that you did. It's not going to be one linear path.
No, not at all. No, I'm glad you're asking these questions because if you look at the Western education system, and I'm not talking about that's the best system. There are many things which is
there are pros and cons.
There are pros and cons of every system. But one of the things which was very obvious to me when I went to Australia was people were joining medicine with arts background. That is just unheard in our country. And if you look deeper into it, it's the training which happens at undergrad level or even at school level. We need to train our students more into critical thinking, problem solving, so that they can move from one field to the other based on their interest. And if you think about when you are, 16, 18 years, 20-year-old, you yourself don't know what is your passion, what you're really interested, except for
Yeah.
few. So if we equip our students where they can move from one field to the other, they should not be limited. Oh, I did my undergrad in biology, so I can't do coding, etc. And as you see, most of the people are scientists who do really well. They have this, interdisciplinary switch very easily. They can understand, and if you look into it, they know how to solve the problem.
Yeah.
They have learned how to learn.
Yeah, I agree with you. But I want to add that in my mind,
Yeah.
the conversation at a 16, 17-year-old, maybe a little later, till 20,
Yeah.
is a conversation that a child must have with his parent.
Mhm.
Sort of a negotiation that let's stop thinking in terms of lanes.
Oh, yes.
Because once you think that this is an electrical engineering lane, this is a biology lane, this is a biotech lane, this is a bioscience lane, this is a biomed lane. I mean, what is this? Like,
No, I think it's changing now, but still it's at a very early stage. We really need to make the young generation feel, hey, this is something you are currently studying.
yeah.
You are getting knowledge, you are getting experience, but this can be translated and you can adapt to different fields and environment.
Yeah, but also like at a very practical level, if you hire someone for one skill,
Yeah.
and then you tell the person, say 3 months down, that hey, I want you to do this other thing, and that person raises his hand and says, oh, I'm not trained for that, I won't do that.
We have few people, and we also have people where I hired somebody to do something based on their CV or whatever
Yeah.
we applied for. Then you realize they are good at many other things.
Yeah.
Usually in 3 to 6 months, we have found people with those sort of skill and we have moved them to that. And even when my research associates come to the lab, they always ask this question, so what project I'm doing? And I'm like, first 3 to 6 months, just get confident in whatever is happening.
Yeah.
And don't limit yourself. If you're from biology background, just look at the wet lab.
Yeah.
Go and talk to the engineering team who is sitting, because you never know who is good at what.
And also, I feel like for them to join your lab,
Yeah.
which is a world-class lab, they also have to have a mindset change that, hey, I'm here in this really world-class lab, and I first must understand how this whole thing works.
So that mindset change doesn't happen immediately. It's a long process.
Okay.
Because they also come from this rigid system. Okay, I
Yeah.
need a project, and most of these guys are looking, okay, I need to finish this project. Either I want to apply for master's or PhD program, which is fair enough.
Yeah.
But in what field would you apply for? What's your career path, etc.? When they are with us, I would say anywhere from 1.5 to 2 is when that sort of
Opens up.
enlightenment happens, when they see engineers talking about tissue and biology people talking about image processing, etc. Then they
Yeah.
are like, okay, I can do this too.
Yeah, and the curious mind wakes up.
Yes.
Yeah.
So it's not an immediate process that happens as they get exposed to, and that's something very nice when somebody comes to me and say, what you have given me is good, but I want to do this, and we are very open to it.
Professor, one question that I have noted here is how do these people who have joined your lab, how do they find you? Like, or rather, maybe, how do you find them?
So, we use the, putting the ads out, job descriptions, etc. on social media, LinkedIn, and on our website.
That's for research associates?
Yeah, research associates, computational, most people. We definitely get a lot of people through our personal connects. Somebody has worked in this lab, and now we are 3 years old, and we have.
You have a reputation. So yeah,
more than
find you.
reputation, now people are slowly getting to know what we do. There are still few people who don't know what we do. So we really want to expand and get really great minds, great youngsters.
Do UG students apply for internships or things
Oh,
like
yes,
that?
yes. We do get, now we are getting quite a lot of students from within Chennai and near about areas, but also now we have got few from north. So slowly, as people are getting to know, we do have students who come here anywhere from 4 weeks to 3 months internship, and the word of mouth, then it goes
I
around.
think for students here
Yeah.
now in the Chennai area,
Yeah.
there's so many really cool internship opportunities. When I was a student here,
Yes.
I didn't have this.
No.
And the visibility I had
Yeah.
was so low, and you
Yeah.
always felt like, oh my god, I have to get out of here.
Yes. No, so that way we are open, but we definitely, I hope even this reaches to a lot of youngsters and they write to us. They can go on our website or they can even send an email directly on my email account, which they will find on the website. And, we are really open in hiring great group of people across the different disciplines.
Okay, I'm going through my notes and I realized that one thing we've not spoken about, it's sort of sitting aside. I was searching online, which is, I use Perplexity because, I wanted to say I use Perplexity because there's an IITM co-founder in Perplexity. So, did you know that? One of the Perplexity co-founders was an undergrad here.
Ah, okay.
So
okay.
We have a little bit of bias towards Perplexity.
Okay.
So, I was plexing it.
Yeah.
And I realized that at some point, Brain Center came up in an Nvidia announcement.
Ah, okay.
And there's a presenter, she takes about 2 minutes to explain, saying that, we are collaborating with this Brain Center at IIT Madras in Chennai. And she talks about something called a Neurovoyager. I'm curious because we spoke about Dharani and we spoke about Atlas.
Yeah.
We never spoke about the Neurovoyager. So what is this thing? And my bigger question was, why does it need a collaboration with Nvidia?
Excellent question. So as I said, we are digitizing these data set at very high resolution. We spoke about 0.5 micron. And if you think about 10,000 of these slices, each one digitized at 0.5 micron resolution, when the entire whole adult brain data is digitized, we are talking the data size as few petabytes. Now, how do you manage these large data sets? How do you visualize? How do you perform different analytics? Because it's not just the size, the speed at which we are generating these data sets. And as I said, our goal is to get the 100 human brain digital data sets out soon. That's where Nvidia come into picture. The tools and the computational tools which they are providing us is really helping to put all of this together.
Nice. It goes from a biology engineering problem to a data science problem.
Yes.
And I can imagine what you're saying. If I'm a data researcher, if I'm a brain researcher and I'm accessing your data and I'm looking at slices, and if it takes me like half a day to just look at the slices,
that's of no use.
Yeah.
You are better off just coming and asking me here. Yeah. So that is something, and as I said, hence as this process of generating these digital data sets got standardized in a lab, our computational work has expanded again exponentially because these data sets are unique. These are at very high resolution and very large data sets, which in a neuroscience community, literally every brain slice could be a PhD project. There's so much of information there. But how do you get all of this information? How do you do this analysis? That's where you need very strong, robust, reliable computational tool and visualization tool. And that's where Nvidia comes into picture. Sorry, you asked about the Neurovoyager. So if you go on our website, Neurovoyager allows you to explore all our data sets, but also query questions related to a particular brain condition, which then uses data sets or publications or information which has been available online.
Oh, right. So I could just type saying, show me this.
Yes. And it also gives you access to a lot of not currently with data which we have not released. You can access those.
Okay. Cool. This is amazing. So the interdisciplinary team has engineers from mechanical, computational, electrical, life sciences, people from life sciences background, neuroscience background, radiology background, image processing background, technicians of all the equipment you use, plus you have collaborators with hospitals, you have medical collaborators, you have research lab collaborators. So this is a huge effort.
Massive.
Thank you for speaking to us. I just want to close a little bit by learning about Dr. Jay, who's your co-conspirator per se, right? What is his background? How did he come
Jay has got very similar background, but he has spent more time in neuroscience. He used to work on monkey and cat brains, and he's really developed as a computational neuroscientist, working with the center again he actually moved here 2016, so much longer. And so He comes with the skill of understanding the brain, but also understanding the computational tools and techniques required to do these sort of analysis.
I got it.
You got it?
I hope the audience follows.
Yes.
Okay, Dr. Richa, thank you so much for joining us. It was a pleasure.
Thank you first for having me here. I'm not a person who goes often, or rather hardly any time on social media or anything. So this is a big step for me, but I'm very passionate in trying to attract young minds in our country to come and work with us and put this word out, the type of work which is happening at the Brain Centre. So thank you for giving me this opportunity and looking forward to having more of these sessions.
You're welcome.
Thank you, Amrit.
Okay, so there you have it. Please like, share, subscribe, comment below. And if you want to reach Dr. Richa, please look up the Brain Centre website and contact them. Thank you so much.